I would like to begin by greeting everyone present here tonight. My name is Margarida Carriço and I am the project manager. We're going to start this session by briefly addressing the two entities behind this project. I will now pass the floor to Carolina Guedes, CEO of Lisbon PH.
Hello everyone, good night. Thank you so much for being here in this webinar with Lisbon PH and NCS. I would like to go through a little brief presentation of Lisbon PH. Lisbon PH is the junior enterprise from the Faculty of Pharmacy of the University of Lisbon, and we are involved in a junior enterprise movement, this world and movement that promotes the approximation between the youth and the young people and the entrepreneurship and all of these essential skills for our future. We are involved all around Europe with more than 370 junior enterprise, and in Portugal we have 25 junior enterprise going through 17 educational institutes. About Lisbon PH, we are the first and only federated junior enterprise in the healthcare sector in Portugal. We are a non-profit organization founded and managed only by a group of entrepreneurial and dynamic university students who focus on the development of projects focused on the accessible training for the healthcare professionals. We act to a clear vision for the development of the health professional of the future, entrepreneurial, creative, and multidisciplinary, guided by 7 values that represents all of our excellence and professionalism. We have more or less 60 members so far and have collaborated with more than 80 clients, but mostly we have trained so far with 10 years of existence, more than 45,000 healthcare professionals. We do this based on our 3 services: events, e-learning, and consulting and logistics that we'll present next. And we have collaborated with more than 30 healthcare companies, and our average client satisfaction is something that really makes us proud. This is a webinar, so thank you so much for being a part of these metrics. This is a type of events that we do online to mimic the presential ones and still have the same information and scientific value that we want to present all of the healthcare professionals with it. So we do this based on our personalized interactive platform, directly streaming to the YouTube. And thank you so much for being here. We also have 4 e-learning courses right now where I would like to focus mainly on the Job Ready e-learning. It's a €10 e-learning course that focuses on all of the essential skills you may need to get your biggest opportunity in the market sector. In our consulting and logistics service, we mainly want to be the solution to all of our clients' needs based in innovation, quality, flexibility, and also, but mostly, a competitive price. We have also a podcast out of the Health Box. This is the Lisbon PH podcast, where we monthly launch 2 episodes focused on the healthcare area, but also with a lot of entrepreneurial topics that might interest you. We have more or less 60 episodes. And we are also present in a lot of different social networks: Facebook, LinkedIn, Instagram, YouTube, where globally we can count with more than 19,000 followers. And alongside our mailing list, where you can receive all of our Lisbon PH activities and news, where we have more than 20,000 contacts, more of them, mainly 60% healthcare professionals, but also a 30% percentage of students. We also focus so much on sustainability, so we have the PH Plus program, our sustainability and corporate social responsibility program that is focused on 3 main pillars: health education, solidarity causes, and also ambiental responsibility. We have so far been able to impact 16 out of the 17 SDGs established by the United Nations, with more than 160 projects and social responsibility actions. If you want to be able to go in touch with Lisbon PH and receive exclusive offers, follow closely our work and be the first to enjoy all of our projects, be sure to scan this QR code so you can enter our newsletter. Thank you so much and have a great webinar.
Thank you so much, Carolina. Next, I pass the floor to Dr. Ana Kadit, founder of the Non-conformist Scientists.
Thank you so much, Margarida. I have here my presentation. It's a great pleasure to be here today with you. This is the 2nd webinar that the Non-conformist Scientist is organizing in collaboration with Lisbon PH, which is very close to my heart because myself, I'm a pharmacist. So I have here the QR code. I'm going to show you at the end as well, but if you want to start scanning it, then you can join our newsletter to know what we are doing. My name is Ana Cadete. I'm a PhD. I'm the founder and president of the Non-conformist Scientist. I live and work in Boston. I'm a scientist. And you can follow me on Instagram at NC Scientist to know about what we are doing, not only with the project NCS, but also where I share some of my insights on the way women in science work in their careers, more recently in maternity, and also where I share insights for scientists in general, and literacy in health and science as well. So welcome to the Non-conformist Scientists, known as the NCS. We are currently a nonprofit organization based in Boston, in the US, with a local chapter in Portugal. We are a team of 16 Portuguese women working around the globe in different parts of the United States and in Portugal and Europe, and we are united by a vision to empower scientists, no matter where they live, to perform at their best. We believe that by sharing webinars and by sharing knowledge like this one you are going to see, that we can empower scientists with the tools that they need to progress and innovate in their work. We are working on 3 major projects. The first one is She Has a PhD, is a podcast, is available on Spotify, YouTube, or wherever you get your podcast from. Take an insight. We are sharing stories of women scientists in leadership positions. And why do we believe this is important? We know that women frequently don't get into C positions or executive positions. So we want to make sure that the young generation of girls in science get motivated by these stories and believe that one day they can also be on the top position in the scientific career. The second project that we are working on are our webinars, as the one you are seeing here. These events are tailored for global education. So we have a network of extraordinary people from professors and entrepreneurs and scientists working on great companies and universities. And we want to share their knowledge with you. That's why we are organizing these webinars and make them accessible to everyone. And then we have the Career Hub. The Career Hub is where we want to partner with institutions or people that are experts in the career field to help the development of career tools for scientists, because we believe that having a PhD is just the foundation of your career. And by having access to different types of tools, then you can actually grow as a scientist and decide what you want to do with your career in the future. As an example, we are launching the Career Path in Science. This was the previous event that we did on April 23rd. And these webinars are intended to share the different opportunities that you have with a PhD that are not only going to lead into an academic position. So stay tuned to what we are doing. Follow us to learn what's the next Career Path in Science webinar that we are going to present. And just before ending, I want to introduce the team and leave here our email in case you want to collaborate with us. So events@ncscientist.com. I'm the founder, but we have an extraordinary team of women behind this. Without them, this would not be possible. So I just want to acknowledge them for making this webinar a reality. Scan the QR code, join our newsletter. Bi-weekly, you will receive our news on the podcast and on the events that we are launching. And I hope that you enjoy this webinar as much as we did putting it together to you. Welcome, and thank you so much.
Thank you, Dr. Anna. After introducing the 2 entities that have made this series of 3 webinars possible, I am pleased to present the theme of our second webinar, Frontiers in Aging Principles of Longevity. Introducing this topic, we have Dr. Claudia Cavado, an aging group at CNC, Center for Neuroscience and Cell Biology, and Dr. Joris Delin, research group leader in the Max Planck Institute of Biology of Aging. Thank you so much for accepting our invitation and sharing your knowledge. Additionally, I invite the audience to raise any questions they would like to see addressed on this topic in the chat. These questions will be presented after exploring the theme. I now yield the floor to each of the speakers. Thank you once again, and I pass the floor to Dr. Claudia, please.
Hello. Thank you for the invitation and congratulations for this webinar and all the activities of non-conformist scientists. And I'm very happy to be here because of several reasons. One, I'm a woman scientist. Second, I'm also from pharmaceutical science. And third, because I'm science and I love to share the science that we do here at University of Coimbra. So if you let me share my presentation. I hope I can be, wait. I know it should be share screen. Wait. I know it's not okay till now. Is it okay now? Is it okay? Someone can? Hello? Is it okay? Was it okay? Yes, I may continue, sorry, because I cannot see this. Okay, just, so I'll try to be brief, 15 minutes to 20 minutes to share what we have been doing at University of Coimbra, especially at the Center for Neuroscience and Cell Biology, and also as a professor at the Faculty of Pharmacy. So the question that you asked me to talk about is how can we influence longevity? What are the molecular and physiological mechanisms underlying aging? But also I'm going to show you why and how are we studying aging, especially neuroendocrinology, and why neuroendocrinology. So as you may all know, the aging is in fact a progressive decline in several physiological functions. So there is a molecular, cellular, tissue, and organism level. And there is a problem in different functions like metabolism, cognition, reproduction, sleep. And in fact, aging is the highest risk factor known for most of human disease like cancer, diabetes, cardiovascular disorders, and neurodegenerative disorders, as some examples. And we know that life expectancy is increasing in all countries. In Portugal, we live 25 years more if you compare 1950 to 2021. So I think we all know that. So we are living longer. And it's also, as I said before, aging in fact is a risk factor for almost all the diseases. And this, I think this data can show you very well. This is in UK. But anyway, so if you look for the cardiovascular disease rates, when we reach the 45 years old, there is really an increase for males and females. And also dementia, around 70 years old, there is a real increase in the prevalence of dementia, and also in cancer. Here the numbers are different, but after 50, 54, there's really an increase in the risk factor of cancer. But so, if a baby born today in Portugal, around here, we have a maternity nearby, so she will live around 80 years old, or a little bit more, 82. But the problem is the moment now, 25% of the years, it will be with a lot of disorders, dementia, at least one or two or three. So these age-related disorders. So the aim, I think, all of us that are working on aging and trying to better understand how we can reach longevity is to try to shrink this number of years. So maybe we will live more, maybe we'll live until 80 or 100 years old, but really small number of years with age-related disorders. So we want to have more healthy life years. So we aim to have healthy longevity. So when we talk about doing research on aging, that people normally ask me is, but do you want to study how can we live forever, live longer, live healthy, and as normally I say, healthy longevity. That's the thing that we want. So how can we achieve that? Is there any strategy? When? For whom? So in fact, I'm going to share the role of science, fundamental science, that can give you some answers to these questions. And now we know very well how the cells get aged and the organism. So it's sometimes some controversial, but we know that when the time goes by, so there is in the cells, in the organisms, there is a lot of hallmarks of aging. So like I'm going to chronic inflammation, there is exhaustion of stem cells, so we have less stem cells and we get age. There are cells that become senescence, so they stop dividing and start to release factors that will promote aging in the other cells around. There is less autophagy, or at least disabled autophagy, this mechanism that is cleaning the cells. Mitochondrial dysfunction, telomere attrition, so a lot of mechanisms that we know very well what is happening now. So this was the role of this. We know we have this information because of fundamental science that gave us this information and much more. And so in my lab, so we aim to investigate the mechanisms and strategies that delay the onset of aging and, of course, age-related diseases. So focus on neuroendocrine mechanism and also peripheral brain axis and aging. So, of course, we want to know how can we increase healthy longevity. So, the first thing so it was the first question: is there any strategy that we can increase healthy longevity or longevity? So we know this is a question that everyone wants to answer a long time ago. So it's not a new question. So, and we know for a long time that caloric restriction is in fact one of the strategies that can postpone aging. So caloric restriction is a moderate reduction in caloric intake compared with ad libitum diet, and it's known, at least from 1935, it was the first paper that showed it in rats, so in rodents. So here we have the age of the animals. Here we have the survival. So in the beginning, all the animals are alive, but the time goes by. So the animals here, they are the ones that can eat the food that they want ad libitum, and in the end, so we have 0 animals surviving. So it means that they died because they are old. But these investigators, when they put the animals, rats, in a caloric restriction diet, the animals live longer. So this was really the first paper showing this, at least published. And so in that time, it was not known what this happened. So it was not very interesting for the scientific community. But now we know that in different models in the lab, so we can have exactly the same effect. So this curve of caloric restriction, so they live longer. We can do it in flies, in rats, in mice, in all the experimental models that we can have in the lab. And of course, there was also, we are not flies, we are not rats, but we are more similar with monkeys. And in 2009, there was the first paper showing that when the investigators put monkeys in caloric restriction diet, so they live longer, but more important, they have less diseases related to aging. So this was the first paper. There was another paper that the results were a little bit different or similar, it depends the way. So the monkeys didn't live longer, but also they have less disorders. So, and there are a lot of information about caloric restriction. This is another paper, an old one in 2009, that showed that women in caloric restriction, 30% caloric restriction for 3 months, they have an increase in verbal memory. So these are information, but how does it work? How does caloric restriction increase lifespan and delay aging in these animals? How does it work? So now we know there's a lot of papers showing that in fact there are a lot of these hallmarks of aging can be reduced in this caloric restriction strategy. one of them is increasing autophagy or modulation of neuroendocrine system or reduction of inflammation. So in my lab, we are trying to tackle this aging thing, having the hypothalamus, is the brain area hypothalamus as a musa. And because hypothalamus, in fact, is like a sensor and a transducer, so it sends hormones, nutrients, and other things. And there is the function for feeding behavior, circadian rhythm, sleep. And so there is really a relevance in this, the role of the hypothalamus. And it's known also that maybe aging influences the function of the hypothalamus. So we started a long time ago with this peptide. It's a neuropeptide, right? It's an endogenous molecule that I was working for in my PhD. And this is an endogenous molecule that is in a high quantity in the brain, but in old rodents, it was demonstrated that the levels of this NPY is decreases. We call it NPY for the neuropeptide Y. And so, and it was known also that this caloric restriction, when we put the animals in caloric restriction, there is an increase of neuropeptide Y in the hypothalamus, or NPY in the hypothalamus. So, this information is important to promote feeding. So, and we could see this also in our lab. So a caloric restriction increase NPY in the hypothalamus. So it was when we start our lab working on aging, we have this information. And so as I said before, so the lower levels of NPY in the brain of old rodents. So we put this question. So we know that caloric restriction postpone aging, but at the same time, increase levels of neuropeptide Y. So our question was, so is there a role for this peptide in aging? Does NPY delay aging? So we started with this very simple question. So for that, we started to work on the autophagy, so this is one of the hallmarks of aging. So autophagy is like cleaning the cells. So there is like the cleaning of when there is aggregates of proteins or organelles that are not good. So there is like the Pac-Man of the cell. This is autophagy. So we put cells, so we culture neurons from the hypothalamus in vitro. Then we put neuropeptide Y, and we could really see that neuropeptide Y increase autophagy. So normally in aged cells, the levels of autophagy is decreased. So this is a marker of autophagy, this is green dots. And here is our positive control rapamycin. So we could see that NPY increase autophagy, so one of the hallmarks of aging. So we published this paper, it's more complicated than that, so it was our first paper related to neuropeptide Y and aging. So NPY stimulates autophagy in hypothalamic neurons. So, but it's not aging, it's not just a hallmark of aging. So then we use animal models of aging. So in collaboration with Carlos Lopes-Otin from Spain. So we use this animal model that in fact they are premature and accelerated aging phenotypes. So they live very shortly. They are low body weight. They have other problems, but in fact they don't live long. So the first thing that we addressed was to evaluate the levels of neuropeptide Y in this hypothalamus. As I said before, all the animals, they have the lower levels of NPY. Here also the same. So it means that this was a good model for our question. So we tried to establish the levels of NPY in the hypothalamus. So these animals, they have lower levels of NPY. Let's see if you reestablish the levels, what happened. So we did that using viral vectors encoding for NPY. So we promote production of NPY only in the hypothalamus, and then we evaluate what happened to the animals. And we could see that there was an improvement of memory. So I'm not going to put all the details, but they became better. But we are not manipulating memory brain areas, but only the hypothalamus. And they can have more fat. This is important in these animals because they are lipodystrophic, so they don't have fat. And even if they eat exactly the same quantity of food, so the skin became, so we ameliorate skin aging markers in these animals, and they look much better, as you can see. So reestablish of hypothalamic NPY delays aging phenotype. Then, so this was in animal model. Then we move to another model of aging. This is very severe. This disorder, Hutchinson-Gilford progeria syndrome. It's a progeria. It's a very rare genetic disorder where the children, so they become with this aging phenotype very early. So they have skin and bone aging, lipodystrophy, so they don't have fat, cardiovascular dysfunction, and the average life expectancy is around 15 years old. Now there is a drug that can postpone until 20 years old. So this is an autosomal dominant disease, so it's a genetic disorder with a de novo point mutation in a gene that codifies for the envelope of the nucleus. So normally, so the nucleus of a cell, of a normal cell, is like this, and in a progeria child cell looks like this. So there is a production of a protein that they called progerin, that in fact that there's impact in the function of the nucleus. So, and these cells, they have all the hallmarks of aging. So it's like a model of aging, and they have this progerin accumulation. So our question was, does NPY rescue or delay aging markers in skin cells from progeria patients? So we did that. So we put incubate cells, fibroblast skin cells from the patients with neuropeptide Y, and we could increase the autophagy, as we saw in neurons from the hypothalamus. We could see that NPY ameliorate the nucleus. They became more round. NPY could decrease this toxic protein in the progerin levels of this protein in these cells. NPY decreased DNA damage marker. It's another marker of aging. And NPY decreased senescence marker. So the cells that became senescent, like zombie cells. So when the cells were incubated with this peptide, we have less number of cells with this age marker. So in fact, we could see in vitro that NPY ameliorates aging phenotype of progeria syndrome cells. So we published this some time ago. So we also saw some of this amelioration also in natural age fibroblasts of healthy men, not in progeria cells. So it means that neuropeptide Y, it was not expected. So we tried to say to the scientific community that neuropeptide Y is an anti-aging player. In fact, we could demonstrate that. But this is just a piece of the puzzle of this of aging. Is this translational? Can we now inject NPY in the bloodstream and can increase longevity? In fact, not, because we know that neuropeptide Y is a peptide, and if we inject in the bloodstream, there will be enzymes that we cut. There is no effect at all. Even we tried that, but it doesn't work in mice. So then we, knowing very well the information and the function of the hypothalamus, we know there is another way that we can increase NPY, and we were inspired by that. We can increase neuropeptide Y in the hypothalamus by ghrelin. Ghrelin is an hormone that is released from the stomach, and so it promotes feeding. It promotes so you want to eat more, and it increases NPY. So this, in fact, can be in the bloodstream, and it's not cut by enzymes. So we did exactly the same in progeria cells. So we incubate the cells with this ghrelin, this hormone, and evaluate the aging markers. So, and in fact, I'm not going to show you the details, but in fact, ghrelin, this hormone, could really ameliorate all the aging phenotypes of progeria cells. From progeria patients. Then we did an in vivo study injecting ghrelin in this the mouse model of progeria. So these animals are very similar to the ones that I showed you before. So, but they live very shortly. They have reduced lifespan. They have cellular reduced progerin. So this is really the animal model that mimics progeria. And so we inject ghrelin every day, daily, subcutaneously, just for 6 weeks, not for the whole time of the animal, the lifespan of the animal. And we could see that the animals They don't eat more. So, and even eating the same quantity of food here in green. So this is this here in yellow. So the animals with progeria, they became with low body weight. But when the animals were injected with ghrelin, the body weight did not decrease as expected. So these are the treated animals. And also we could see there was an amelioration in the progeria mice in the skin of the animals, comparing ghrelin with vehicle. And there was a very relevant amelioration of adipose tissue. So this is the adipose tissue, if you remember histology. This is the adipose tissue, the adipocytes in wild-type animal with no disorder. This is in the progeria mice. They are really very small, so they have almost no fat at all. But when the animals were administered with ghrelin, so we can really revert this lipodystrophy, even they eat exactly the same. So, and also important to could see in different tissues the decrease in this protein, this progerin, this toxic protein. So, and more important, so the animals live longer when they were injected with ghrelin. So this is the paper that we published last year, so showing that ghrelin delays premature aging in this syndrome of progeria. Then just only more 2 minutes to show that there is other things that we are doing in the lab, especially related with aging. We are looking for the importance of sleep and circadian rhythm. And because when rhythm dysfunction can increase aging and dysfunction. So we are looking at for in the very not important disorder, very prevalent, but we know the people normally don't make the diagnosis, that is obstructive sleep apnea. And we are proposing that this disorder can accelerate or may accelerate aging. And we have now in the lab a lot of information showing that. This is one paper that I'm not going to show that. So we are now working also a lot in the related aging and sleep. Also another project we are doing now, we were inspired with this paper that shows that mortality is written on the face. So it is a very, very nice paper, but we propose another thing. We are proposing that maybe the skin is not only the mirror of our aging, but also increase our aging. So it's like a trigger of systemic aging. So the question now, just to finish, is healthy longevity in our hands? What should we do? And I think all of you know the recipe. So healthy eating, the quantity, quality, and the hours of eating, quality of sleep, stress reduction, avoid smoking and excessive alcohol consumption, maintain social relationship and intellectual activities, primary health care through lifespan. So it's not only preventing aging when it starts to show the aging phenotype. Healthy environments, of course, including air, soil, and water. Stop discrimination. I'm not going to explain, maybe you can discuss it after. And of course, fundamental and research, and applied research is important. And physical activity, I forget this one. So I'd like to now acknowledge everyone in my lab and thank you for your attention. I stop sharing, hopefully.
Thank you very much, Dr. Claudia, for enlightening us on this theme. The topics covered were indeed not only interesting, but also really important to address. I now pass the floor to Dr. Joris de Lien. So please, when you can.
Thank you for the invitation. First of all, it's really nice to be able to speak to you. It's a bit late, but that's fine. I will manage. First of all, I would like to thank Professor Cavadas already for the nice introduction, which helped me to go to it a bit quicker, because actually the first slide I had was indeed the same, saying that the aim of our studies is no longer to really extend the lifespan of individuals, but really to look of ways of extending the health span. So this is what the future will hopefully bring for us. We managed very well to extend lifespan, but now the time has come to extend our health span as well. So I want to talk a bit about two different topics that we are studying in my lab. at the Max Planck Institute for Biology of Aging in Cologne. And the first one is on the biomarkers of healthy aging. So, as you may know, people are always looking for good markers that can indicate how healthy somebody is at a current age, so that you don't need to wait and see what happens in 10, 20 years when it comes to diseases. So there are several criteria that people have come up with, which should be met by biomarkers of aging. And one is obvious is this: it should outperform chronological age in predicting age-related disease and mortality, because aging itself, so your chronological age, is already a very good marker of how healthy you are and how long you will live. But the idea is that these biomarkers would be even better. It's also very important that they are simple and inexpensive to test. So, which means that ideally in an individual, you can just take a blood sample or fecal sample or something where you can easily and cheaply, hopefully, test this without harming the subject. And another thing is that it should work in both humans and model organisms, so that we can actually translate the findings between the two. However, disappointingly, we don't think biomarkers fulfilling all of these criteria will exist. So we try to do our best to come up with markers to fulfill at least two of these criteria. So, and over the last couple of years, there has been a massive shift in the field towards these markers that are based on larger data sets, such as epigenetic data, it can be proteomic data, metabolomic data, and really come up with these predictors of healthy aging, or so as people would like to call it, biological age. And the idea is that people have been training, they have taken these measurements and look how well they predict chronological age. So they train predictors on chronological age, as you can see here on the right side. And then once they have done that, they go back to the individuals and they actually look, okay, you have now your age predicted on the marker that we were measuring, which can be, for example, epigenetics, and we have your chronological age. And now we look at the difference between the two. And in the case that your epigenetic age is higher than your chronological age, we say that somebody can be epigenetically or biologically older. And the same applies the other way around as well. And the idea is then, if we then take this difference between your biological age and your chronological age, that is a marker of how healthy you are. However, you can also use other kind of methodologies where you not train the predictor on age, but use other outcomes, which we think is actually a bit better. And we did this as well in my group, together with many collaborators, especially from Europe, where we actually tried to create a predictor of mortality. And to do this, we took a metabolomic platform, which measured many different metabolites in the blood of an individual. And then we identified actually 14 different markers that are predictive of mortality. And when we combine them into one model, they independently predict mortality. And these are markers such as your polyunsaturated fatty acids, lipid-related markers. There are also several metabolites like leucine and valine and isoleucine. There's a very interesting marker over here, which is an inflammation-related marker. And what this plot indicates, if the levels are going down, it means that an increased level of those markers is associated with an increased chance of dying. Well, if we see, decreased chance of dying. But if we see it over here, the marks over here, an increased level is associated with a decreased risk of increased risk of dying. And when we then made the score and checked it among all the populations that we had, we see that it's a very good predictor of mortality. So it predicts like with a hazard ratio of 2 to 3, which means that one standard deviation increase in the score gives you a 200% increased risk of mortality. So it's a quite strong marker. So we wanted to go. So different groups have done different things, which are pretty similar for other types of markers. Or for epigenetic age, for epigenetics, for example, there's this score called GRIM-Age, which is also trained on mortality. And what was very interesting for us is that a completely independent group of us actually started to test also our predictor of mortality, which we now call Metabo Health, in the different populations. And it actually shows that this predictor that we created, which is trained on mortality, is also a very good predictor of incident diseases. So I don't know how well visible it is, but over here you see a different kind of scores they tested in the study, and the Dale and All score is the Metabo Health score that we created. And you see that it associates with mortality, cardiovascular disease, type 2 diabetes, COPD, dementia, and cancer. So it's really not only a marker of general mortality, but also of general health, which is very nice that we have that now. And what was also interesting to see is when we did a study where we took the metabolomics-based biomarker of mortality and one that was based on epigenetics, and we combined them together, they independently predict mortality, which means that we can use different kind of measurements and combine them to actually improve even the prediction of mortality and also morbidity. So what are we doing at the moment with this score? So we actually are very interested to see determine if the score can complement existing measurements that are already used in the clinic. So in the clinic, for example, people make a lot of use of frailty to estimate vulnerability of individuals or single markers, and we want to see, can we bring this marker that we created into more clinical settings? Another thing is that we would like to know how heritable it is. So is it really driven by your genetics, or are there more environmental factors that drive the decrease and increase in this score? And this is very important because it could indicate that we can come up with good lifestyle interventions, for example, or pharmacological interventions to actually target the metabolites, which would improve the score for individuals. But if it's more genetically driven, we need to think about other types of approaches. So this is also directly related to the next point, that we actually want to see if specific interventions can also influence the score. And we want to see how we can further improve the predictive ability. So we know that it associates with mortality, but we really are interested to see, okay, what kind of additional markers can we add on top to really improve even more this prediction and come up with a better marker of general health. So in conclusion of this part on the metabolites is actually that we have a set of these 14 metabolic biomarkers that we created a score out of, which is very predictive of all-cause mortality. And we see that it's actually working even better than conventional risk factors, something I didn't show today. And really, we're trying to see, can we bring this metabolic health score now into a more clinical setting? And really, our goal is that you would come to your general practitioner, you get your metabolic health measured, or something that is based on that, and that can actually say something about your current health instead of the standard questions that a clinician is currently asking. So we really want to see, can we implement this in also hopefully clinical decision making in the future. And then I want to completely shift the focus to the genetics of human longevity, which is actually the main thing my group is doing. So what we know about longevity, and longevity is here defined as living to an extremely old age. So say, for example, 90 years of age or 100 years of age, also a bit depending where you're living, but really this extremes in the population. Because we actually know that this is a heritable trait. And we also know that many people that live to such an extreme age are also living more healthily. So if you would do it on general lifespan, this is not the case. But if you really go to these most extreme individuals, you see that these individuals have a delayed onset of many age-related diseases, and even sometimes a complete absence of those. However, we know that it's likely heritable. We don't know the exact estimates, but we think this heritability is strongest in the individuals that belong to the 10% longest lived of their specific birth cohort. So if you take an individual born in the 1900s, you look in all the people that were born at that time, and then you only take the 10% longest, and this is really inherited as a heritable trait. However, we also know that longevity is assumed to be very complex. So we don't expect that there's one gene explaining longevity completely. We actually expect that there are many different genes that could actually make you live longer. And it's even more probable that it's a combination of different genetic variants or different genes that work in concert. So there are different approaches in which you can use to actually study genetic variation, and I will shortly touch upon the one that I have done since my PhD and we started with, which is mostly focusing here on the common genetic variants. So these are variants that are relatively common in the population. So at least 5% of the individuals in the population has them. However, we then expect that the effects on the specific outcome are often a bit smaller. And when we did this, actually, when we compared this very long-lived people to the general population and looked for variants that are different between them, we and others did many different studies on those. The only thing that consistently pops up is this ApoE protein. And there are two different variants. Oh, sorry, and this is in populations from European descent, but also from East Asian descent. And when we zoom in a bit on this and look, okay, what are the variants in this gene doing? There's actually one variant, which is the ApoE4 allele, which is associated with a decreased probability to become long-lived. So as you can see over here, this odds ratio is below 1. And we see this very consistently across populations. And very interestingly, this is also a gene that was already found to be, or this genetic variant was already found to be associated also with increased risk of cardiovascular disease and increased risk of Alzheimer's disease. So it's really a disease risk locus. However, very interestingly, there's also another variant in this gene, which is the ApoE2 allele, and this is showing the opposite effect. So if you carry this allele, you have an increased probability to become long-lived. And actually, this is also associated with a decreased chance of developing Alzheimer's disease and cardiovascular disease again. So this is more of a so-called disease protection or longevity locus, which we are hoping to find. However, as mentioned in the table that I showed over here, there are not so many variants that we identified. So actually, I decided to change focus and no longer focus on this common variants, but look now for variants that are maybe unique for these long-lived people or very rare in the general population. So the hope is that these long-lived individuals carry specific kind of variants that we do not see in the general population. However, it's very difficult to study such variants because it's often not possible to determine causality. So if these people have them, they have many rare variants. So how can we actually determine if those variants are actually responsible for the longevity of these people? If you test these common variants, you can do statistics. You can say, okay, this 5% versus 7%. But in this case, this is not possible. It's 2 people versus 0. You cannot do proper statistics on that. So we really need to do functional studies. And we can do that with cellular models, and we can do that in model organisms. And to really narrow down the list of variants that we can look at, we normally focus then on variants in genes or in pathways that have already been associated with lifespan regulation in model organisms, with the idea that we will also see the same in humans. So this is my team working on this. So I have at the moment 3 PhD students, Larissa, Mara, and Tolua Mimi, 1 postdoc, Maruf, 1 former PhD student, Helena, that I co-supervised, which also worked on this, and then my 3 technicians. Actually, I have 4 technicians at the moment. Andre is not shown here, but Alina, Sarah, and Ramon. And they're really needed because especially the mouse studies are quite big that we are doing. And I'll come back to that in a moment. So the idea is what we want to do. We want to, first of all, identify genetic variants in these long-lived people and then really perform in vitro characterization of them. So for the selection, how do we do the selection? Well, we do whole genome sequencing of this very long-lived people. So we sequence their whole DNA and we really try to focus on special individuals, namely long-lived families as much as possible. So these are individuals where there are multiple people in the same family that have reached at least an age of 90 years of age. So at least 2 people in these families belong to the 10% longest lived of their birth cohorts. We also use so-called singletons in part of our studies, which are these individuals that have no known history, family history for longevity, but they were actually all above 95 years and even above 100 years. But the majority of the work is focused on these types of variants. We then select them. So how do we do that? We focus on the genes that I mentioned before that coming from this longevity studies of model organisms, and then we look in the pathways that have been found there and look there for genetic variants that are then protein altering. So we know that they influence the structure of the protein, and we also use for that this CAT score, which can tell you something bioinformatically that it's very likely that these variants influence the functioning of the protein, and they are absent or very rare in the general population. And the next step is then that we generate transgenic cell lines and mouse lines using CRISPR. So our focus has mostly been on the insulin IGF-1 signaling pathway. So as mentioned already in the previous talk, actually caloric restriction also worked through this pathway. So this is a very important pathway that has been found to be manipulatable in different ways, pharmacologically, genetically, in model organisms, and this often leads to lifespan extension. And we often see that this goes to this mTORC1 and mTORC2 complexes, especially the mTORC1 complex. We think it's very important when we target that complex and we reduce its effect, that then actually that could lead to the lifespan extension, because this influences the downstream targets such as FOXO. I'm not going into detail. If you have questions about it, feel free to ask afterwards. But what I want to show here is that we generated different kind of cell lines with these mutations that come from these long-lived people, and then we looked at this downstream target. So we looked at the downstream target of mTORC1, which is phosphorylation of S6, and we see clearly a downregulation in the cell lines, in most of the cell lines that we have created. If we look at AKT, which is actually a target of mTORC2, we don't see this so clearly. So we see some of the cell lines that do not have an effect, and some show actually an upregulation. There's also another target, which is in a related pathway, which is called the MAPK-ERK pathway. And there, downstream of that, you have the phosphorylation of ERK, which is also a marker from model organism studies to be associated with lifespan regulation if it's downregulated. And again, if we look in our different variant lines that we have created over here, all of these lines also show a downregulation of this MAPK-ERK signaling. So this indicates that we think that these variants potentially have an effect on cellular health by downregulating these two proteins, phosphorylation of these proteins. What was very interesting for us as well is that when we created a transcriptomic and a proteomic data set based on our cell lines, we see a very clear clustering of the cell lines. So we have a group over here which contains 3 different cell lines, and the different symbols are the different isolation rounds. And here we have another group which contains about 7 cell lines, where we can clearly see that they cluster away from the wild type cells, and they also cluster away from each other. So there's 2 different things happening here. First of all, they are different from the wild type, and second of all, they are different from each other. And we are now really trying to figure out what these profiles indicate. We do not know yet, but we think that this might be 2 different ways by which, well, the variants could lead to cellular health and potentially even the extended lifespan in their carriers. But that's a long shot, of course. Another thing that we notice is that it's not only on this proteomic level, but also when we look at things like stress resistant, but also at growth rate, we see that this grouping is also represented. So, for example, if we look at the growth rate of the cells, you have here 3 of the cell lines in greenish that are coming from one of the groups, and the 2 in blue from one of the other groups. And you can see that the one from the group over here, they grow faster, and the one from the group over here grows slower. So this is the confluency rate, and you see over time, these go faster and these grow slower. And this is also actually, when we go back to the data that I showed before over here, we actually see the same. So this was this phosphorylation of AKT, where also this one from one of the groups actually show an effect and the others do not. So we actually see this grouping represented all over the place. And we are really trying to figure out what that means now. We also looked at in vivo characterization in mice, which is currently ongoing. And what we do there is we check the lifespan of these mice. So first of all, I'd like to say that we managed to create 4 different lines of the mice, 3 of the variants in the first group and 1 of the variants from the other group. And those we are characterizing. This is a lot of work, so we cannot do this for every mutation that we have. So we do a lifespan, we check the lifespan of these animals, we do phenotyping at 3 different time points, at 4 months, young age, 15 months, which is around middle age, and a bit older age, around 18 to 22 months. And during this phenotyping, we measure an array of phenotypes, including frailty of the mice, the gait speed, so how fast they can walk, how good they can walk, their grip strength, also their body composition, and things like glucose and insulin tolerance tests, because the variants that we are looking at are involved in insulin signaling, and we expect that those things would also be influenced. And we do that then 3 times during their life. So we have just finished for one line the last round of phenotyping, but I will not show the data today due to time restrictions. I just focus on one phenotype after this, which is the lifespan. And then we also do from all these mouse line tissue collection. So we collect also tissues at the same time points from a different cohort of mice. And the idea is we do not only do this in the normal state, but we also stimulate the mice by starving them and letting them refeed, so that hopefully stimulates the pathway. And this is still work in progress. So at the moment, like I said, one line, we have just finished the phenotyping. We are currently in the process of analyzing the data. We see some potential interesting phenotypes, mostly already in young age, not so much in old age, but in young age, they might show some beneficial phenotypes. But the most interesting for us is actually the lifespan. And what we see at the moment, we cannot say anything yet about if something is statistically significant, because we have to wait till they all die, which takes still quite some time. So this one is the first, but even there's still around 20 to 25% of the mice alive. But what we could see is already for 2 of these lines where we have the lifespan data that's far enough, that if we look at our homozygous mice, which are here indicated in dark blue and here in dark purple, they seem to show a slight tendency to have an increased lifespan. And especially here in this line, this seems to be mostly coming from the young age. And when we looked into these mice and we look what they die of normally, this is something which is called ulcerative dermatitis. So this is a disease of the skin. Their skin gets infected and then they have to be sacrificed at one point, or they die very quickly. And we see that our mutant mice seem to be a bit more protected from this. We have to look deeper into this, but this seems to be a quite clear phenotype that we can observe. The question is now, of course, how does it run when we wait till the end? But this was already quite interesting for us, given that you have to be aware that these mice just contain one single genetic variant in their genome that is changed, and we already see differences. So this was quite interesting for us. So in conclusion of this part, so we know that the genetic component of human longevity is complex, and it's likely determined by many different variants, which are probably rare. We already successfully generated the transgenic cell and mouse lines carrying these variants that we identified in long-lived individuals. We see that most of these cell lines show changes in the expression of the genes which are harboring them, and also alter this insulin IGF-1 mTOR signaling. The different transgenic cell lines show this overlapping transcriptomic and proteomic profiles, which we are currently trying to unravel. And we already see some promising beneficial effects on the health span of the mice. So of course there are many people that contributed to all this work. So these are my international collaborators, especially the work on the metabolomics, and the earlier large genetic association studies was done by me in combination with a big consortia. So this is really a lot of people that were involved, and this is only a small part of those. And then of course specifically also the people in my current group that do all the work with the mice and the cells, and my collaborators from the Leiden University Medical Center and Kiel University that allowed me to work with actually the data of their long-lived individuals, and then of course the core facilities at our institute, which are fantastic, because without them this work would not have been possible. And last but not least, the funding agencies for supporting my work. Thank you for your attention.
Thank you so much, Dr. Joris and Dr. Claudia, for your clarity on this fascinating and so special subject. We will have now a roundtable discussion where I introduce the moderator, Dr. José Pedro Castro, invited professor to teach biogerontology and nutrition at aging at Faculty of Medicine at University of Porto and Nova Medical School. He will lead this conversation when we will have a more in-depth exploration of the topic. At the end of this session, I will present the questions added to the chat, so I encourage you to input any remaining doubts. Thank you for your collaboration in this webinar. So please, when you can.
Right. So can you hear me? Yeah,
Yeah.
probably yes. So first of all, and before we start the discussion, I would just like to congratulate the organization for this beautiful event. I think it's pretty clear that longevity is currently the hot topic, and myself and Joris and Claudia that have been working. I don't know. In my case, for 15 years, it's great to see how the longevity field is kind of being recognized and show the importance across borders. So yeah, first of all, thank you for organizing this, and you're doing such an amazing job. And then I mean, this is kind of new to me to be kind of moderated, so we will try to make it flexible. We had two brilliant presentations, and it's good to see that people are kind of exploring different facets of aging. It's good to have different interventions and people thinking differently. And so I congratulate Claudia and Joris for that. Right now, we don't have questions from the audience, so I'm kind of gonna kickstart this conversation by asking you two a very general question, and before we kind of move to very specific ones. And Claudia, maybe for you, and then maybe Joris can answer this as well. So we have been working a lot in the aging field, and now we're kind of witnessing a lot of money being injected in the field. Many people are involved, many labs, but we still haven't found the cure for aging, let alone what biological age is. So what do you guys think that it's currently missing in the aging and longevity field?
Thank you, Jose, for this difficult question. So, in fact, the first thing, I think we still need money. So it's not enough. If you compare with the United States, for example, we don't have in Europe NIA agency to fund the aging. So I think we still need money. And also, this, as we said, when we started working in this area, it was not very fancy. I think it was not very attractive. And now it's amazing to see how the students are really interested in this area. So I think now we are getting more people with a risk of having also people that are more predatory. So it means that people that are entering in the field not by the science area, but more by the commercial thing. So this is a threat for the field. So I think there is missing. So the models of studying this is always like in other, as in aging, but also in other disorders like neurodegenerative disorders. So we use, for example, rodent models. There are some problems also. So I think the models are. So we still need to
right.
Get,
yeah.
I think there is I think we are going to increase we need more people, and this is very multidisciplinary. You need more people with different approaches. So I think it was nice to see how these two conversations, the two talks were different but complementary, and I think and we need.
Yeah. And I just before Joris speaks, I would just like to emphasize precisely that, because as soon as you start working on aging, you start seeing how complex the thing is, and you're starting to study one tissue, and all of a sudden you're studying a different tissue, and then you're using bioinformatics, and then you're using something else. So it's kind of a really complex puzzle, and it's great to be studying this at this point. So Joris, sorry, I interrupted. You probably want to jump in.
No, I can jump in. No worries. So there's actually one important thing that's also missing, which hasn't been mentioned, is we don't actually have a fundamental understanding of aging yet. We have the hallmarks, but the hallmarks are not the fundamental understanding of what aging actually is. So, and I completely agree with this point that we need more diverse people in the field to also approach it from a different perspective. I think this is what the field is still a bit missing. We really need to have people from other kind of fields jumping into it, say from a more physics background or mathematical backgrounds. Those people should jump into the field and help. And it's what's indeed very interesting to see. I'm already in the field since my PhD. when the field was on a high and then became low again due to some controversial findings. Now we are up again, but we have to be careful that we do not run into the same risks again. So we shouldn't overhype. We should be clear about what we know and what we don't know about aging. But there's a lot of potential. I mean, this is clear. Investors are super interested in it. People always get excited when we talk about it, but we have to be careful that we don't hype things too much, because if they don't work, this will definitely influence the field. And so we need more people, more money. I completely agreed with that, and hopefully get a more diverse representation in the field as well.
Right. And I think that these hallmarks were kind of really important like 10 years ago, that showed that the field is not abstract and we can interfere. But as a conceptual framework, I think we need a little bit more, and we need to look under besides the lamppost, right? And okay, and so I really started this bad as a moderator. Apparently, there's a lot of questions from the audience, so I need to apologize to all the listeners. And I would just like to start with, well, if there's time, I will ask you some questions, but I would like to encourage and kind of read the audience questions. I think it's more important that we kind of give them this spotlight. So Tania Medeiros, she asks, well, she says, thank you so much, Professora Cavadas. How does NPY stimulates autophagy? Does ghrelin affects NPY directly or indirectly?
Okay, thank you, Tanya. Great questions. In fact, so we investigate the effect of NPY in autophagy in hypothalamic neurons, and in fact, NPY stimulates two receptors, and we know exactly the pathways involved. So it's related to PKA, ERK1/2, and AKT. So we know it's mTOR independent in these cells. But not in fibroblasts. So it depends on the cells, the effect, so the mechanisms are different. When you ask the other question, does ghrelin affect NPY direct or indirect? We didn't investigate that in the work that I showed you here in progeria cells, but in fact we investigate that in neurons from cortex from the brain of the rats. And we could see that there is in fact an effect of ghrelin through NPY. So there is a crosstalk between the hormone and the peptide. So thank you. Great question.
Yeah, and I think now we should move to the other one, and it's from sorry, my screen is so small. It's from Diana Rocha, and now it's for Joris. Am I pronouncing your name correctly? Is it Joris or Joris?
Both is fine. It's Joris, but that's harder to pronounce, so it's fine.
So
my last name is more difficult for people.
Yeah. Okay, so she asks, Diana, how can genetic biomarkers be used to predict an individual's risk of age-related diseases such as Alzheimer's disease, cardiovascular disease, and even cancer? I think that's a very good question.
Exactly, that's a very good point. So first of all, what we know is we don't have many variants for longevity yet. But what we can actually do, and this is also what we did in one of our studies, you can look at the so-called genetic correlation. So you take all the variants across the genome and you look at their correlation with longevity, and then you also look at their correlation with specific age-related diseases. And then you actually see that there's a very strong inverse correlation between longevity and cardiovascular disease and longevity and Alzheimer's disease, but not, for example, between longevity and specific cancers. So although we know that there is a correlation, but it's not so strong. And we know that aging is also important for cancer. Cancer is not the strongest age-associated disease. This is very clear. But then also, I have to say, also type 2 diabetes is a very strong correlation. And very interesting, if we actually look at lifespan, so not at longevity, but just your general lifespan and do genetics on that, then we see a stronger correlation with cancer. So we think that this longevity trait that we are studying mostly is something special, which is the avoidance potentially of cardiovascular disease and Alzheimer's disease and type 2 diabetes, but not so much of cancer. And this is also represented if you look at these people themselves, because there are quite some long-lived people that still developed cancer, but they
Right.
overcame it. So they developed cancer during their life, but it was not the thing that killed them in the end.
Right. So in the end, what you're kind of telling us is that that those variants are probably related to particular trajectories and not other trajectories, right? So maybe cancer is not one of those trajectories more common to those particular variants, but Alzheimer's is okay. So because there's this, now we know that there's these very different trajectories, right? People age very differently, and you might get cardiovascular disease, some other people might get cancer. So and I think this is really important what Joris is telling us, to kind of understand these differences in trajectories. Great. So I'm sorry about my voice. I'm a little bit sick today. And now moving to Rita Almeida. She asks, I would like to ask, how can genetic testing and personalized medicine approaches be used to tailor interventions for healthy aging based on individuals' genetic code? So it's kind of a personalized medicine thingy. Thank you, Rita. Really nice question. I think.
Good question. So what I would like to start off with, which I realize I didn't tell you, is that only 15 to 20% of longevity is determined by your genetics. So most of it is actually lifestyle. So lifestyle changes will definitely for sure have the biggest effect, much bigger than genetic things that we are doing. If we talk about genetic testing and personalized medicine, this is really tricky because it's just a risk, right? So what we can say, okay, even if you have the ApoE4 allele, right, this negative allele that I was talking about, it doesn't mean that you have a 100% chance of developing these diseases. So you can tailor your lifestyle based on what you hear that you get genetically, what your genetic risk is. But I would anyway tailor my lifestyle already into that direction. For example, people that have ApoE4, people say, oh, you should eat less cholesterol because it's a cholesterol binding enzyme, protein, and that it's involved in the transport of that. So they say eat less cholesterol, but this is anyway healthy. So you could anyway do that without knowing if you have ApoE4 or not. So I think we shouldn't emphasize the genetic test because they really only work for the things where it's very clear that a variant leads to a disease. So there are some cases like a BRCA1, BRCA2 for breast cancer where it's very clear if you have that allele, you should get something done because the risk is the penetrance is super high. But for the variants that we identified for longevity, but also for other diseases, it's not so clear. So I would just anyway change my lifestyle and think first about my lifestyle and personalizing that before I would look at my genetic risk and take that into account.
Yeah. And I think that this can also lead to lazy people because, they can be tested and they have like protective variants and they say, okay, I don't need to do any exercise. I can drink, I can smoke, I'll be always protected. And we know that's not the case, right?
No.
So thank you, Joris, for telling us that. And now I think we can move to the next one. And I think this could go to Claudia Cavadas. She's very, involved in this type of things. And the question comes precisely from Anna Cadets. Will this be on the screen? Otherwise, I can just tell you. So bridging the gap between science, medicine, and society, how do you see this research helping us live longer and healthier lives? Do you have any practice advice for the attendees? Because most of the audience will not be aging researchers. So that's a very good question.
Yeah, I think this is a good example. This webinar that we have to talk about science that can explain how caloric restriction works. So I think we need to have more science literacy and health. And so the literacy in science, it's important that people understand better. When we talk about autophagy, it's nice that we can see that someone can understand. So I think we have to continue to talk about this fundamental and basic science related to aging. This is one thing. And of course, the other things that we can do that was already, we also talk about that. So 70% is related to lifestyle. So I think I used to say, we know already the recipe, but science can explain. But I think we have to be more careful about the sleep. So the physical activity, social interaction. So we know the recipe, but science can explain. So I think for some people, it's when we see, we have to sleep more, they don't care. But if we explain the science behind that, so it's more convincing for some of them. So I think we can. That's why all of us can do something related to our health and the people that are around, and science can explain. So this, if I can, this is my answer.
Yeah, and taking a little bit on that, we now know, for example, that sleep deprivation increases biological age and inflammation. And if we know how, for example, the transcriptome changes, and we know drugs that will, kind of reverse these signatures, these transcriptomic signatures of inflammation, for example, we can, based on science, kind of, develop drugs that can kind of ameliorate all these bad things that we know happen during aging. So this is just a kind of a side note. And so, there's a lot of questions. I don't know how we're doing on time. I think we have kind of 10 more minutes or so. And so I'm going to keep reading the questions as they come, as I see them. And I think the next one is again for Claudia, but Joris, please feel free to, if you find you should talk about interventions. We try to make it dynamic. So the question is from Lucas Faria, and he says, "Hello, I would like to ask, what are the potential therapeutic targets within the neuroendocrine system for promoting healthy aging and preventing age-related neurodegenerative diseases?"
Yeah, so there's a lot of intervention. So because if you think about neuroendocrines relating hormones to the brain, so the hormones, we have to keep the hormonal things good. So when all the exercise, all the healthy lifestyle can be, it's a strategy. So I think this in the aging field, I think it's, for me at least, it's difficult to, of course, I call it neuroendocrine strategies, but we should have really an open mind with all the mechanisms, because the aging field or the aging strategies is, we are not going to have like a target, the target. So that's why I think caloric restriction is really relevant, because it touches different targets. So saying this, so I think neuroendocrine strategies, even sleep is a neuroendocrine strategy, because it interferes with the hormones. So I can. I don't know if Joris, you want to add something. Even it's not your field in neuroendocrine, but if you think about hormones and other.
Well, your point, which is very important, we don't expect it's one target, right? So we expect it's very complicated, and there are many different ways. Which you also think from the genetic side that can target aging. It might be represented at specific types of profiles, but we don't expect that it's just by changing one target. It might help a little bit, but if we really want to make a big impact, we need also different targets, and actually also the how they work together, which is also very important and very complicated.
Right, and I think that's why systems biology became so critical in the last decade or so, because it's showing us how little we knew about aging, so how complex the puzzle is. But now we have better tools to kind of understand how the cells talk between each other, and how tissues crosstalk, and so it's a really complex. So I agree with both of you. There's probably not a single target. Well, it might be that a particular trajectory of aging might be more relevant for that particular target, but as an overall, we don't think there's one target of aging. I think this is a misconception, and I think this is kind of ruled out currently. Right. So before we move to a more activistic question, which I think it's pretty cool, I have one question from Joris. Sorry, Diana, for jumping, but I'll come to you in a second. And the question is from Jessica Reis, and she says, "Thank you for the insightful presentations." So for both of you, of course. So she asks, Dr. Joris, longevity is intrinsically linked to heritage, or there is investigation in mimetic the epigenetic outcome. So I'm not
I think I understand.
yeah,
I think you understand what she's aiming at. So like I
Harry.
said, it's we think it's around 15%, 15 to 20% is genetic. So it's not linked to her heritage, and we actually think that the environment, which is often reflected in the epigenetics, is a way that we can actually also target aging. What is very interesting, of course, is that if we look at these very long-lived people, that some of them were genetically predetermined, they sometimes have super unhealthy lifestyles. So, I mean, they have probably something which makes them very special. And if you have those types of genes in combination, you might indeed not need all these lifestyle factors.
Mhm.
But in general, we also think that even in those situations, this good lifestyle factors play an important role. And I mean, the blue zones, don't know how many people have heard about this. This is also now on Netflix. It's a very interesting concept that you have this place in the world where people live very long and healthy, and this they can trace back that it's potentially due to the environment and their lifestyle habits, but also part of it is genetic, because we also know if you take those people out and put them in somewhere else, they can still have a slight advantage. But if you put people from outside in those blue zones, there are not that many studies, but there are some. They don't live so long. So it's also this gene environment interaction, which makes it even more complex. So we have the
Complex.
genes, we have the environment, and we have the interaction. But we think that the environmental component is the biggest. And regarding the epigenetics, this is also reflected in epigenetics. If you smoke, it will change your epigenetics, for example, and this will have a long-term consequence. So yeah, and diet is the same. So this is something that we can work with, and that helps us also in research to come up with interventions.
If
Yeah.
I may jump in this
Sure,
question,
sure.
because it's very
Yeah.
nice. Because there is also other studies showing, not in our field, in more less biological field, but the people that have a purpose of life,
Yeah.
they live longer. So this is
Mhm.
the social
Motivation. Yeah.
interaction,
Mhm.
the motivation. So it's something that is behind even the environment and the genetics. So I think, and this we cannot, the biology cannot explain yet. But the purpose
No,
of
it's
life
Right.
is incredible.
Yeah, people that
Right.
live. So one of the main risk factors of people dying at old age is actually loneliness and depression, and this then influences their health in their body. So it's but it's hard indeed to quantify how much it is and to study biologically. But in this blue zones, it's also an overarching concept that those people have indeed a purpose or meaning in life, and this is probably also playing a role. Yes.
Yeah, and also the social interaction. If you compare the longevity of people from Portugal and people in Spain, I don't know now the last numbers in longevity, but Spain was really the country the poor longevity. Sometimes after Japan, and I think they don't eat very well. The sleep is not so comparing to Portugal. I think it's the social interaction. They go out in the intergenerational communication. I think this is
So the message is, if you don't have time to sleep, please be with your friends.
yes.
That's the great, great. So yeah, so moving on, I think we're on our last minutes, but I would like to ask just a few questions from people that I didn't have time to go through, and one is from Diana Roche, and I think this is kind of for both of you. And she asks, what are the societal implications of an aging population, and how can we adapt healthcare systems? That's a really critical point, and social policies to support healthy aging.
You want to
So it's
start?
yeah,
And,
fire away.
I can start. I can start. I think it's sometimes that we should be careful with this. We saw this in the during COVID. That the old people, this is a problem. This is an economic problem, and in fact, if they live healthy until the end, there will be not a problem. So, and if you see the Japanese society, they have a lot of old people, and they don't have so much problem. So that's why I think health literacy, uh, healthy aging, healthy lifestyle, it will be the success of our healthcare system. And we need to have more people studying aging and gerontology and geriatry. So, we should avoid this. I would say this. Ageism, thinking that the old people will be like it will be like a problem. It's not a problem. That can be very important in the society differently. So we have to think differently. This is
And they
my.
have so many stories and so many experience of things, right? So we can learn so much from older people. So I think this is a very important question. But sorry, Joris, I interrupted you. You were going to say something.
Yeah, I would say completely agree. And one thing, if you can think about how we should reform our healthcare system, is actually to focus more on aging and not on the single diseases. I think one problem of our current healthcare system is somebody comes in, they check, oh, you have a cardiovascular disease, they treat you for that, and they send you out. And we need to go more into this treating aging. If it's a disease or not, that's another discussion. But at least that the people come in and that you can look at the more holistic picture and say, okay, we are not treating the one disease, but we're treating the people differently so that people age healthier. And then I completely agree, if we do it right, people live healthier for longer, and we actually need less healthcare. This is the idea behind it. There are other problems that we might get with overpopulation and all these kind of things, which is a discussion not only for aging, but for other diseases as well.
Yeah.
But there's a misconception that aging is only bad. But if we treat it well, then it would actually become beneficial for everybody.
Right.
Yeah, we should invest in preventive medicine. So we should prevent,
Not react.
not
No, exactly.
So it should be preventive. This
Right.
is
And the data actually supports that, right? This work from J. Oshlansky in the 90s, that it shows that if we cure all cancers, we are going to have a 3% benefit in lifespan or health span, because some other disease will take over. And so instead of treating one disease at a time and kind of pushing the healthcare system, we should treat aging to compress all these chronic diseases. So I absolutely agree with you guys.
Let me just say something. That's why I think it's important that we talk about longevity instead of aging, because longevity is more positive, and there is less. So I think.
And it's better to get grants if you talk about the longevity than right. Okay, so thank you for. I think there's one more question. I don't know. We don't have time. I had a lot of questions for you guys. We don't have time to go through them. But there's one question here from Ricardo. I think it has to be the last one. And he asks, and I think this is for Claudia. He asks, has NPY been studied in other animal models, probably meaning like physiological aging or something, to understand if some of these mechanisms are evolutionary conserved?
Yes, so when it was in our lab, so we have been working also in neurodegenerative disorders, and NPY can has a protective role. So in Machado-Joseph disease, so there is other, also in the retina, in epilepsy, so there is a neuroprotective role. So this is the only thing that I can say very shortly.
Right. And I think the question is really good, because just last year we published with my former lab, the Gladishab lab, we published in Cell one paper kind of understanding what are the longevity signatures that are favored by evolution in long-lived species. So it's kind of, we're shifting fields as well towards understanding what nature has gifted animals, long-lived animals, to have this really long lives and longevity. So this is one of a hot topic right now. People are trying to understand that, because if we think about it, like lab rodents, they are very useful, but they just live 2 or 3 years max. So there's a lot of mechanisms that evolution did give them, that we need to kind of untangle to use in our favor. So I just would like to finish by asking you two, what are the major milestones you kind of envision for the longevity field in the upcoming, let's say, 2 to 5 years? Is that a reasonable? What do you think is going to be the real, the biggest milestones in the longevity research?
So I think the biggest milestone would be, depending on how it goes, that we can show that the drugs and things that have been identified in model organisms can also work in humans, so that we come with a drug intervention that extended lifespan is something that we cannot test, but at least show signs that it can slow aging. That I think that is a realistic thing. If it's 5 years, we have to see, but I think this will be a very big breakthrough if we can show that the things coming from the model organisms will actually work in humans as well. Which drug it will be, we have to see, but I have high hopes that there will be at least something that will do that.
Yeah, I also agree. Maybe I can add another way also, if you can make really rejuvenation with the reprogramming cells. So there is already some in vivo work showing the reprogramming cells that can rejuvenate some organs. Maybe that is also an important mechanism of information. I don't know if it will be like a drug, but I also agree with Joris that will be. We will have for sure 1 or 2 drugs. Maybe we have already and we don't know.
Yeah, it could be.
Exactly.
It could be. Right. From my side, thank you so much. It was a pleasure moderating to really, amazing scientists in the aging field, and I hope to see you soon. And now I don't know if the organization has to, wants to give like a final word or something. So, but thank you.
In this manner, I bring the second webinar to a close. I hope it has been enjoyable for the audience. To our speakers, I hope you have enjoyed collaborating with us as much as we have enjoyed listening to your expertise on this topic. I'd like to draw your attention to the QR code present on the template displayed on the screen. By scanning it, you will have access to the satisfaction survey for the session. I appreciate the response from all the participants so we can improve our services. Lastly, I would like to mention the next webinar in this series titled AI in Drug Delivery. This event will take place on May 16th at 4 p.m. Eastern Standard Time, 9 p.m. Greenwich Mean Time, and 10 p.m. Central European Summer Time. We will look forward to your presence. I now pass the floor to Dr. Tanya Medeiros. Thank you so much.
Thank you so much. I hope you like this webinar as much as we did. And I would just like to inform to join our newsletter so you can scan here the QR code and to be updated with everything that we do at NCS. Thank you so much for joining.